Okay, so we're going to start tonight with a slideshow about Wildlands Trust, just a brief introduction to our organization. And I know a lot of you are familiar. I see a lot of familiar faces. Thank you all again for joining us. Wildlands Trust works throughout Southeastern Massachusetts and the 4 counties to preserve and permanently protect native habitats, farmlands, lands of high scenic value and ecological value. And we do this in order to keep our communities healthy and our residents, you, connected to the natural world. That is our mission. This is our service area. So we're in over 40 towns across Southeastern Massachusetts, which makes us a regional land trust. And there are regional land trusts across the state doing similar work, and so that the whole state is covered. I think there's 70 some odd nonprofits in Massachusetts. We're a very good state for nonprofits. Actually, it was started here by the trustees of reservations in the late 1800s. So go Massachusetts. Our work kind of falls into 4 categories. We have the land protection. So we have about 10,000 acres in our holdings right now for Wildlands Trust across the region. And once we have that land, we steward it for the public and wildlife and clean water. We have a whole crew that's out there constantly keeping the trails safe for the public to use. And then we have educational events and community engagement programs. That's kind of where I come in. So events like this, we do, in normal times, we would do lots of outdoor guided programs. Right now, we are offering some outdoor seated programs where people can safely social distance. So we have yoga and meditation happening at our headquarters in South Plymouth. And we're also offering these online Zoom programs in place of our in-person presentations during this time. And finally, I always like to give a nod to the towns, the other nonprofits, and the state agencies in our service region, because our work would not be possible without those partnerships. Where you come in is our members. So as a nonprofit, we are member supported and donor supported. If you're not already a member, you can join for just $50 a year, and that $50 goes directly towards our mission of conserving more land in Southeastern Massachusetts. You also get discounts to any paid Wildlands Trust programs, so those yoga and meditation workshops I was talking about, members get discounts to those, and you get discounts to local farms and retailers across the region that have partnered with us. So I will be sending everybody a follow-up email to today's program with lots of information on ways that you can stay involved and learn more about cyanobacteria, but I'll also include a membership link in that email. So that brings us to tonight's program, which is Understanding Cyanobacteria in Our Ponds with EPA scientist Hilary Snook. And before I turn it over to Hilary, I'm actually going to turn it over to our partner organization, the Herring Ponds Association, who are co-sponsoring this program with us tonight. By turning it over, here we go.
Thank you, Rachel.
Yes, we have Don from the Herring Ponds Watershed Association.
I'm Don Williams, the president of the Herring Ponds Watershed Association. Next slide. And I just wanted to give a brief overview of what the Herring Ponds Watershed Association is. Our ACEC, or Area of Critical Environmental Concern, constitutes 450 acres, 11 ponds. Our main focus is on 3 of those ponds, Little Herring, Great Herring, and Elbow Pond. And our mission statement, simplified for this presentation, is to preserve and protect both water and land. And we are people. First and foremost, we are all an all-volunteer board of directors, and we have over 250 members and over 400 on our mailing list. Next slide, please. We were founded in 2007. We've been around for a while. We've been monitoring our water quality continuously since 2008, and we have formed a very good partnership with the town of Plymouth during this time. Next slide. Our accomplishments in a nutshell: educational newsletters. My wife does those, and the presentations that we've been doing most recently with Wildlands Trust. We do a herring count every year, except this year. We've printed in our 2nd edition now the stewardship guides that advise people what some of the chemistry and biology of the watershed is. We do E. coli testing at 11 of our bathing areas. And I'm putting this in advance. We're going to beat a cyanobacteria bloom. We've helped Plymouth purchase a number of recharge lands in our area. And with Plymouth, we've remediated 3 runoff sites. So we're an active group. Next slide, please. And then there was Hilary Snook. Now, Hilary is an EPA Region 1 environmental chemist. I met Hilary first at an APCC seminar on Cape Cod several years ago, and then attended with my wife and a couple of members of our Watershed Association, a Cyanobacteria workshop at North River Watershed Association about 2 years ago. Next, please. So what happened to us? On July 17th, the world fell apart. We had a cyanobacteria bloom in Great Herring Pond for the first time in anybody's recent memory. Next, please. What happened was a resident took a picture, sent it to the Plymouth Health Department. We got an email that Friday afternoon from David Gould, who is our contact at the Plymouth Department of Marine and Environmental Affairs. The health department closed us down. And as I said, this was the first ever cyanobacteria bloom at Great Herring Pond. Next, please. And then there was Hilary Snook. I mean, this all was coming down on us during a weekend time. Hilary was around Friday night. He was around Saturday answering questions over the weekend. And my wife eventually sent him some pictures of the cyanobacteria that she took under the microscope that we had bought through his workshop. And he identified it as a toxic microcystis cyanobacteria. Hilary has been great in providing information to us throughout the course of this crisis that we've had, and we are forever grateful to him. We're grateful to Rachel for arranging this so quickly, and to Hilary for being so flexible. And without further ado, I'm done.
So thank you everyone for joining the webinar tonight, and I'm glad you can make it. Hopefully this will be informative and helpful to you guys, and as you tackle the problem of blooms down in your neck of the woods. So next slide. Okay, so today's presentation, what I'd like to do is cover a few different things. You can click through that, Rachel. This is a cyanobacteria primer, just to kind of give everybody a baseline to understand where cyanobacteria come from, what they look like, how they behave, and then progress into blooms, what causes them, and why we care about them, why they're important to us, and then what we can do to mitigate our concerns and the problems, and a little bit on the current state of the science as it stands right now. Next, please. All right, cyanobacteria have been around a long time. They started about 3 billion years ago. You can click next. And we know this because of geologic formations that have been around for 3 billion years. The picture on the lower left, back up a little bit, Rachel, if you would. The picture on the left is a picture of stromatolites from Shark Bay, Western Australia. These geologic formations can also be found in eastern Montana in the foothills, and what they represent is striations of cyanobacteria that they have dated back 3 billion years ago. So cyanobacteria have been around a long time, and even though we're having current problems and woes with them today, they were very beneficial to us as they extracted the carbon dioxide out of the oceans, replacing it with oxygen as a byproduct, and then off-gassing that into our atmosphere, which actually gives us the air that we breathe today. So we can be thankful they were around 3 billion years ago and have been in existence since that time, and now are coming back to cause us a little bit of issues. So compared to the dinosaurs, they've got a much greater head start on how to adapt and survive in the current environment. Okay, next step. They've also caused their problems way back when as well, and there's evidence out there that shows that they produce toxins even in the era of the dinosaurs. And there's been some archaeological digs that have shown pretty convincing evidence that toxic cyanobacteria decimated certain populations in certain areas around watering holes and things where they actually drank the water and succumbed to the toxins that were in the cyanobacteria. Next, please. So where do we find cyanobacteria? Basically, in a word, they're found everywhere. You find them in the Arctic regions, the polar regions, tundra. They can be found in hot mineral springs. They can be found in desert environments, like on the left. And actually, during the first Gulf War, a lot of the soldiers came back with symptoms that kind of alluded to cyanobacteria toxicity. And they actually found cyanobacteria in the desert sands that they think were ingested or respirated into the GIs and caused some of the toxins and effects that they showed later on. They also, besides the detrimental effects, they still to this day have positive effects. That picture in the lower right-hand corner there with the little plant growing, you see it's surrounded by this kind of this purple material. That's actually cyanobacteria in a desert environment that's covered with a sort of filamentous mucous membrane. That actually absorbs water out of the atmosphere and actually helps keep these plants alive and thriving in these arid environments as well. So they're all across the planet in every environment you can think of. But our main focus is the aquatic environments for us tonight. But I just want to make you aware that they are everywhere. Okay. Okay, you've often probably heard of them called blue-green algae, and the question has come up time and time again that I hear, well, are they algae or are they bacteria? And probably the best way I've heard of it summed up is that really they're structurally like bacteria, but they function a lot more like plants. Hence they were called blue-green algae way back when. Now algae is really limited to eukaryotic plants and algae. Instead of cyanobacteria, which is prokaryotic. Next, please. One click. Okay, so if we look at a general algal cell, green algae, it has a very nice structure. You probably remember this from your 5th grade or middle school biology class. You've got a nucleus there. You've got a cell membrane and a cell wall. You've got flagella that gives it some motility and movement. And you have, probably most significant is the chloroplasts. And within those chloroplasts is where you find chlorophyll, which the algal cell will gather sunlight and use that for producing food and energy. And if you compare that to cyanobacteria, next click please. It's a much more simplified structure. You don't have the organelles within the cell wall so much. It is surrounded by a mucilaginous membrane, simple cell wall, and then some of the organs and stuff are kind of loosely assembled within that structure. Probably the most important for us is to look at these photosynthetic membranes, which are actually called thylakoids. These hold a particular pigment very similar to chlorophyll. It's called phycocyanin, and it uses that to photosynthesize the sunlight and use that as energy. So that's where it gets its functionality, very similar to plants. All right, next please. So this is just a schematic of looking at the absorbance of light from chlorophyll and phycocyanin. You can ignore the rhodamine here. It's important, as you see later on through the presentation, but what I want to gather here or portray is that they absorb and emit light at very different wavelengths. Chlorophyll in the 400 range, for those that know, and emits it at 600, and the phycocyanin in the 600 range. So it absorbs at a certain wavelength of light and emits. And that's one of the reasons phycocyanin has that blue-green color to it often, is because it's absorbing in the red light and it's reflecting out the blue-green that's on it, and hence comes the name blue-green algae. So we can use those different absorbance and emission characteristics to really understand and track cyanobacteria better. So one more click. Sure. So we use an instrument called a fluorometer, and as soon as that chugs forward, Rachel, when it comes, there it is. This is the tool that we use for measuring those different wavelengths of light, and we can actually track the green algae that's in the water column as well as the blue-green that might be emerging with this particular instrument. All right, next slide. So a little bit more of the unique characteristics of cyanobacteria. If you look on the left, you'll see the thylakoids that I mentioned. This is the area that houses the phycocyanin, that pigment that absorbs the light and produces energy for the organism. It's in kind of disorganized folds, but again, it's had 3 billion years to evolve. And those folds actually greatly increase the surface area that it's able to absorb light. It's just like folding a piece of paper many times together. You compress its overall volume, but the surface area is quite large. Then it has food storage particles within it, but the other part that I want to draw your attention to is the gas vesicles that you see in the slide to the left. These are very important to the organism because it optimizes where it can be within a water column by adjusting its buoyancy to optimize where it is under light conditions. They actually are sensitive to light and can be destroyed by too much sunlight, so they really have this feature that allows them to optimize that characteristic. If we look on the right-hand slide, you'll see a very common cyanobacteria, and what I'd like to point to is a couple interesting things that cyanobacteria have in many cases. And if you look, you can see this mucilage. It's this kind of like ghost-like sheath around the outside of the bacterium here, and that is a protective layer that many of them have to protect them from the elements, this protective coating. There's also an aconite, kind of a elongated cell, and that is what we call a resting cell. When these bacterium die off, that cell will fall to the bottom of the lake, and it can rest there until conditions are right to reproduce or regenerate. It could lay there for years until conditions are optimal, and it comes up and propagates. The heterocyst, or heterocyte as they are now called, is basically a nitrogen fixing cell. It can take dissolved nitrogen out of the water column, convert that to ammonia, and use that as an energy source. So if we look in the atmosphere, where we have 72% nitrogen is what our air is made up, and only 21% oxygen, it can really take advantage of that. And again, this is through its evolutionary 3 billion years of evolution that's given us these really good thriving characteristics. Next slide. Okay, within cyanobacteria, there are several different forms. We have unicellular cyanobacteria, so these can be small single-celled cyanobacteria that float around in the water column and can be distributed ubiquitously throughout your water body. We also have colonial forms. They regenerate or propagate, and they'll form basically these dense colonies. I'll show you some examples in a minute. And then we have multicellular filamentous types, which grow more in elongated filaments, almost like blades of grass that they will grow. We also have cyanobacteria that can be less than 2 microns in size, very, very, very tiny. If you look at a typical spider web, silk of spider web, that is about 5 microns in diameter across. So these can be extremely small, and that has real relevance to some of the issues going on today, which I will explain as we move further along. There are close to 100 different genera of cyanobacteria that are out there and over 2,500 different species. So that's a lot for a taxonomist to learn and take care of. But really for our purposes, there's really only about a dozen or so genera that we really need to pay attention to within the region here. So next slide. Okay, so what do they look like? They come in many different forms. What I'm showing here is an image-based key that was developed by the University of New Hampshire that we helped them work on many years ago. And we call it the Dirty Dozen key because these are the top dozen cyanobacteria genera in the region that are known toxin producers. So as you can see, just from looking at these different images of these different genera, they come in many different shapes and sizes and forms. Next slide. So I'm just going to go through a couple of the most common ones here just to kind of give you a little perspective. This used to be called Anabaena. It is now mostly referred to as Dolichospermum. This is a filamentous type of cyanobacteria. They grow in these long, almost like beaded necklaces, and they can vary from 5 to 20 microns in size. They contain the heterocytes as well as the aconites, those resting cells. And there's a couple different forms. There's a benthic form, which is found that doesn't have the gas vesicles that promote bloom, so they will primarily be found on the bottom of water bodies. And then there's also planktonic form, which has the gas vesicles, and they can be found throughout your water column, and again, ubiquitously across your water body. And these can be toxin forming. Since they do have the gas vesicles, they can form dense, odorous, and problematic blooms. And the last thing I will say about a lot of the cyanobacteria is they do have taste and odor issues. So this can be a problem in drinking water systems, not only from the toxin standpoint, but also from just the tastes and odors of drinking water by some of the chemistry that they produce. Next slide. Right. Aphanosomina is another common one within the region. These vary from 3 to 8 microns. And this is a filamentous type of cyanobacteria. So the cells are joined end to end in these like barrel-shaped cells, and they grow looking kind of like, as you see in the middle picture there, kind of like little blades of grass that are found in your system. They also contain the nitrogen-fixing heterocytes and the aconites, those resting cells. And they have gas vesicles, so they can be bloom formers as well, floating to the surface. You usually see these in more nutrient-enriched water bodies, and they're usually associated with more than one type of cyanobacteria. Taste and odor issues as well. So next slide, please, Rachel. Okay. Microcystis, this is one of our more common ones that we see a lot of. This is a colonial form, as you can see in the lower right-hand corner of the slide group there, kind of the mucilaginous coatings around the colony, and the individual cells packed within that. The one on the lower left side is actually a slide mounted, so it kind of squishes the colony apart, and you get kind of that lacy-like pattern. So these are also found with others. They do have gas vesicles, so they will float and form blooms. They have quite a bit of protective mucilage around them. Nutrient lovers. And again, most of these cyanobacteria like the warm conditions of the water. And these can be very dense and be associated with other cyanobacteria within a bloom system. Okay, next please. Okay. So most of the limnologists like to call these most dominant types that we see Annie, Fanny, and Mike is what they used to be called. And now that Anabaena is Dolichospermum split up, they refer to it as Dolly and Fanny and Mike. So you can use that lingo and sound like an expert a little bit if you want. So throw that out there for you. Next slide. All right, so what's the drivers of cyanobacteria? Why are we seeing more and more occurrences of cyanobacteria blooms occurring across the country and across the planet, actually? This year we've seen, I've heard of several lakes that have never bloomed before that are now blooming across the New England area, and other lakes that were chronic bloomers that they just expected every year and they haven't seen the blooms yet. So they're elusive and they're interesting at the same time. But if you look at some of the drivers of what is causing this increase in blooms, this is a picture, that little graphic in the front center there is actually from the Atmospheric Administration that shows the change in precipitation patterns through the period of record since the 1900s. And this really is looking at the actual intense rainfall frequency that has occurred. So we're only looking at the heaviest 1% of rainstorms that have occurred through the period of record. And you can see that through the 70s and 80s and 90s and 2000, we have seen a dramatic increase. And most of you are probably well aware, we look at the rainstorms that we used to get and what we get nowadays. We get these intense storms where we're getting a couple inches in just a couple hours, where we didn't usually see that very often. Instead of being once every 10 years or so, now we're seeing it every couple of years. So we're seeing that increase in frequency, intensity, duration, and frequency of these rainstorms. Next click, please, Rachel. So for the Northeast, oh, one more time, sorry about that. Let's see if it comes up. Oh, I lost it. Anyway, the graphic was on the last slide that didn't show up on my other presentation where it fell apart. But basically what we're seeing in the Northeast is, in the last couple of years, a 71% increase in these precipitation increases in that, just at 1%. And there's lots of regional variation where we're seeing some real anomalous changes going on in weather patterns. Another one here, and before I go further, what probably the take-home message from that with the precipitation changes is, if you had a pristine water body that didn't have any infrastructure around it, there are no houses, it was just out in the middle of the wilderness, we would still see an increase in nutrients coming into that water body just by that change in that precipitation pattern. So even under perfectly natural conditions, we would see that increase due to the climate. So if we look at average daily temperature that comes in across the season, we track that through the period of record, you see that increasing climb as well. And that, even though it seems subtle, it has a big change in what's going on within these systems. We have less snow on an annual basis. We have less ice cover. If we do have ice cover, it's usually for a less duration of time. The snow melts earlier than before. We have less of it. All those factors come into play in regards to cyanobacteria issue within the region. Okay, next slide. And if we look at the way infrastructure and our human factors come into play, one thing I like to call watershed dehydration, and we look at the infrastructure that goes on with rooftops, roadways, stormwater runoff. It really has changed the whole hydrology of our systems. Even with smaller developments, we've noticed big changes in rivers and streams at less than 20% development in a watershed. And for lakes, if you think of this example here, which is in the city of Worcester, Mass., with all this infrastructure, we're not getting the rainfall, the snow melt infiltrating into the ground and coming into the lake through groundwater and kind of keeping that pulse moving through the duration of the warm summer months and into the fall. Instead, snow melt and rainfall is going directly into the system and immediately out of the system. So we're not able to keep, it's kind of like strangulating the circulatory system of the water body. So I like to call it, I coin it watershed dehydration, because basically what we've done is taken that water out of the system, shot it out very quickly. In streams, we see much higher flows and much drier periods and much lower flows because of this stormwater infrastructure that's now in place. Next slide, please. So this is an example I like to use all the time because I think it really emphasizes the points I just talked about. I'm sure most of you are all familiar with Monpont Pond and the issues there. Want to click on that, Rachel, again? And one more time. Okay, so issues that are the drivers. Again, we discussed climate change taking place, also the nutrient loading from roadways, runoff, agriculture, lawns and driveways, that sort of thing, the warming of waters. Low flushing rates is another example where, because we're getting that hydrology, it's coming in through storm and leaving so quickly, the lake isn't getting a constant flushing or a re-cleansing of new water coming into the system. And as the lake warms up, we start to get stratification where the upper surfaces warm up, the bottom ones are colder, and the bacteria in the bottom of that water is actually using up a lot of the organic matter that's settling out, and it's using up the oxygen that's down in those deeper depths. And when you use up that oxygen, Basically what it does is it releases the nutrients that are sitting down there bound up in the sediments when there is oxygen down there. So it changes the whole chemistry on the bottom. And then the watershed dehydration that I talked about. So one more click, Rachel. So, oh, go back one for a second. So if we look at Monpont Pond, which I'm sure most of you are familiar with, we have a causeway running between the east side and the west side. Water comes in through the east side of the pond. I think there's only 1 or 2 small culverts that connect the two, and the water leaves out of the northwest corner there that you see in the pond. So it's really, the flow is restricted within the system. It is a shallow system, which has a tendency to warm up quickly. All right, next click. All right, and we do have agriculture on the water body, and cranberries are one of my favorite fruits. I love them, but these fields do use anywhere, because they use up so much nutrients in the growing. Fertilizer application rates are anywhere from 10 to 60 pounds per acre per year on these bog systems, and after they harvest, then they're dewatered for taking care of the plants, and that goes directly into the water body. So you have all these things: low flow, warmer temperatures, a nutrient pulse coming into the system, no flushing rate. And next click. And this is kind of the end result. So I love this slide. It's a great example of all these little things coming together for the perfect storm on how cyanobacteria does manifest itself in the water systems. Okay, next slide. And not to put all the fingers on agriculture, there's a lot that we can do as homeowners as well, and we can point the fingers at ourselves a lot. Natural systems, if you look at this slide, this is from the University of Wisconsin. I thought it was great, so I have borrowed it in many presentations. But if we look at the volume of runoff, the phosphorus inputs, that one really limiting nutrient in lake systems, although nitrogen can play a factor too, and the sediment inputs that go into a water body. Under natural scenario to the far left, a natural system, you see very minimal inputs. But next click, Rachel. As we start developing, we start to see increases in sediment influx. We have closely cropped lawns. Usually that means a shallow root system for the grasses, and that allows more erosion to take place. And then if we have a well-developed waterfront, we really see much, much larger increases in runoff and phosphorus loading to the system. All right, next slide. So on to blooms. What exactly is a bloom? Basically, you hear people say, well, you'll know it when you see it. And if you look at the slide on the right, I don't think there's anyone on this presentation today that would say, nope, that's not a bloom. We look at that as a nice, clean system that I'd want to go swimming in. The definition I've seen for blooms, and they're kind of hard to
it looks like we might be having a trouble with Hilary's connection there. Let's wait for him to come back. Let's see, Dawn, this might be a question that you can answer. I believe it pertains to Herring Pond. Why take a sample at the boat ramp when the site, why take sample at boat ramp when site was opposite side of pond?
Well, the first sample that they took, they farmed it out to somebody from Envirotech, and they took the sample there. It was a weekend, and the guy who took the sample admitted that he didn't even see any cyanobacteria there. So that sample came back 0, not unsurprisingly, but it was kind of a total waste, and really didn't send us in the right direction at all.
Okay.
Because it was early and it was vacation.
It was early. It was a vacation. It was a weekend, and
The town couldn't mobilize somebody
the town couldn't mobilize somebody.
to take.
Do you want to do this?
Yeah.
Good grief.
Do you
So-
guys know if we know the exact species that has been detected in Great Herring Pond?
Yes, we have sent pictures Jerry, my wife Jerry, took microscopy pictures of some of the bloom that we took ourselves and that Kim Tower of the Department of Marine and Environmental Affairs took. Now those samples were sent to Hilary. He looked at them and he said that they were the microcystis. The problem that we're having with the analysis from the lab is that the lab said that there was Anabaena, that there was pseudo-Anabaena, and 2 other types that I don't know at very low levels. And that just didn't make any sense because visually there were a lot of cells going on. And what I'm doing now, just so people know, is I'm trying to work with the town to open this up. I think the burden of proof is really, it's really on the town to let us open. That's the way that it's done. The town goes to the state, they recommend opening, and the town is going to recommend opening only if they don't see a bloom. Now we have a bloom, but very low levels of cyanobacteria. All of the cyanobacteria that were identified are toxic. And the other piece of evidence that we should know is that there was at least 1 person that I know of who was swimming in the pond who was sent to the hospital for 4 to 5 days and was in pretty serious condition. There are a couple of dogs that have gone into the water and have had serious problems requiring veterinary hospital care. So we do know that there is toxic cyanobacteria. We do know that the town has the ultimate say with the approval of the State Board of Health. It's not moving them so far to see that the numbers are low. They are toxic. There is a bloom. That's all they know about. We're trying to get them to move off of that square one because we feel that based on what we've seen, and we're not experts by any means, but my wife has seen fewer of these microcystis in her microscopy. And there seems to be a diminishing as time goes by in the 2 samples that we have. So Hilary's back. I hope that clears things up. It's really confusing, as Hilary will be the first to admit that these things have been around for 3.5 billion years, and they're pretty skillful at hiding their footprint.
Thank you very much, Don and Jerry, for answering some of those questions in the chat. Welcome back, Hilary. I'm glad you made it back.
Me too. I actually plugged in an Ethernet cable. I don't know what happened, but hey, I should have expected it today, right?
Yeah.
Right.
All right.
So,
well, I'll let you jump right back in here. Thank you, everybody.
Okay. So, so basically, like I explained to the bloom, you know it when you see it. It's basically defined as an increase in cyanobacteria biomass that usually persists for a couple days to a couple weeks. It's usually composed of, maybe a couple different species, usually not too many, and it's visible to the naked eye. There are algal blooms that occur, which algae blooms, and these are different, but you can get dense mats of other types of blooms from green algae, which are for the most part benign and not potentially toxic. There is 1 species of brown algae that usually you find in the Southwest Texas area that is toxic, but that's about it, not in this region. But we do have other types of blooms that are non-bloom formers as well, and that's the Cyanobacteria. These are less than 2 microns in size. So again, extremely small. If you took 1,000 of those, you'd span the head of a pin, just to put it in perspective for you. These are often widely dispersed throughout the water column and the water body, and they can be easily aerosolized just based on their size. So they can get into the atmosphere just from evaporation processes taking place on the lake. And these are some that are easy to inhale from skiing behind a boat or being near a waterfall or a spillway of a water body. So there's some concern with these as well. Next, please. So how they form, basically the cyanobacteria will basically spend most of their time in the photic zone all the time. That is the zone that light penetrates down into the water column. So they're gathering sunlight to function, so that is where you are going to find them. Next slide. So usually they're fairly dispersed, but as the water starts to warm up, green algae really don't like it above 59 degrees Fahrenheit. They start to 15 degrees centigrade or so, they start to die off, and that's kind of where the cyanos start to outcompete them. Cyanos really like the warmer water. When it's around 77 degrees and higher, they really start to take off, and that's where they start to thrive and propagate. And their cell divisions can be 3 times per day. So if you think of how quickly that can overrun a system, it can happen very quickly. And usually that happens about the same time that the lake is stratifying. You get the warm surface layers. The lake starts to show anoxia in many cases, and those nutrients that are released from the bottom start to get mixed in the upper surface water into that photic zone. And then you have the perfect storm again, where they really take off because they've got all the fertilizer they need, the nutrients, the warm temperatures, the sunlight, and off they go. Okay, next. Oops, go back one just for a second. So as they start to concentrate in the water column in the photic zone, often during calmer days and cloudy days, they start to move up closer to the surface as well to optimize where they are for gathering sunlight. Okay, next slide. So what we can actually do is we can actually track this progression of when they start to multiply within the system and really start to take off by looking at the ratio of chlorophyll and phycocyanin, those 2 pigments, and predominantly phycocyanin, but we can also look at the chlorophyll too, because in a green system, the chlorophyll will start to die off. As the temperature is warm, the phycocyanin will start to accumulate, and that yellow chart there just shows the quick accumulation of cyanobacteria concentration. The white graph there actually shows the Secchi disk depth, which is basically a measure of the transparency or clarity of the water column. So as would make sense intuitively, as the cyanobacteria start to propagate and they get denser in the water column, your clarity drops. And you can see a significant drop in clarity as this goes into almost an exponential growth curve. And this slide is compliments of Nancy Leland from University of New Hampshire that just works with part of our collaborative. So it's been a great tool to show that. And so we can use that meter I showed you earlier to kind of track that progression, kind of get a heads up on when a bloom might be occurring or might begin to occur. Okay, next slide, please. So you've got the cyanobacteria. It accumulates at the surface under these dog days of summer, these gray days and warm days. It's up near the surface. And then you have a windstorm that kicks in, and it disrupts that surface, and it disrupts the buoyancy of the cyanobacteria. So next slide. So with that, it all kind of pops to the surface because it takes them a while to regulate their buoyancy, which they can't do right away. So it all comes to the surface. And then next slide. You have to excuse my cartoons here. This is about the best my graphics get. So, and then the wind blows it to a windward shore someplace where it stacks up and creates problems and really gets concentrated. All right. Next slide. All right. So this is a picture, again, complements the University of New Hampshire, and it shows the actual phycocyanin concentration transposed to cell counts across a particular water body on a particular day. So the message out of this is it really depends on where you're taking your measurements in your samples on what it might reveal and tell you. If you're in the upper part of this water body and you're in the pink zone, you're going to get 415,000 cells per milliliter. The state cutoff for putting out a public health notice is 70,000 cells per milliliter. Now, if you're down the lower end of the pond, you might only be reading 750 cells. So, as John had mentioned, and I'm sure most of you know, it can be quite elusive, and it takes a little bit of thought and planning on where you do your sampling and how you look at your system. Okay, next slide. Blooms come in many different forms. Just a smattering of them here. If you look at the one in the upper left, that is a bloom that is taking place throughout the water column, planktonic species, so it's throughout the photic zone and evenly dispersed throughout the system. And you can see others. The next one to the right is one that's stacked up on the shoreline, a small bloom. This photo is compliments of Nancy Leland, UNH. The one down in the front center, that's actually a pollen bloom. So that is often mistaken for a cyanobacteria bloom. We often get lots of calls on it, but it's relatively benign. But we have also found cyanobacteria intermixed with these types of blooms as well. So even though it looks benign and we know it's a pollen bloom, that doesn't mean you may not have cyanobacteria in it. The one on the far right, that golden color, and then you see a green underneath it, that's two different types of cyanobacteria, and they kind of formed layers, one on top of the other. So next slide. So when we talk about toxicity of some of these blooms, we can't tell if a bloom is toxic just by looking at it. You really need to do the tests on it to make that determination. The toxicity is not affected by boiling water, so if you're boiling water at camp or something, you're not going to get rid of those toxins if they are in there by boiling it. The death of the cells, we have intracellular and extracellular toxins. So that little graphic I have on the right there, you could just for demonstration sake say that the big yellow ball is the cell, and all those little green things inside are the intracellular toxins, those that are encased within the cell walls. As these blooms wash up on the shore, sunlight gets to them and they start to decay. You can click once more, Rachel. My fancy graphics here. The cell walls dissolve, and then these toxins are released into the water column. And that's often why, I know the questions come up, well, you know, the bloom, we don't see a bloom anymore, so why is the Board of Health closing us down for 2 weeks? And this is the reason, is because you can have a bloom occur, it can die off, and these toxins can be released into the water column, and they can persist for weeks. Usually it's not that long, but a couple weeks is not that uncommon. So that is kind of one of the reasoning behind that, keeping systems closed for that. Okay, next slide. All right. So for those of you that see a bloom and you might be curious if it's a problem or not, there is a very simple jar and stick test that you can use. Click once more, please, Rachel. So if you use a stick and you see some gunk on the shoreline, you can pick it up and it sticks together like on the right. You can be pretty assured that that is going to be a green algae bloom and not a cyanobacteria bloom. The only case where that doesn't hold true is for certain benthic forms of cyanobacteria. You can get mats that will float up to the surface under certain conditions, and you could have a toxic form then. But most cases you're going to see it like this, and it's going to be a filamentous form, kind of a cottony texture. That's going to be an algal bloom and not a cyanobacteria bloom. The image on the left is what we call our jar test. If you put what you find you suspect is a bloom or green water in a jar, and you put it in, put a lid on it, and leave it for an hour and cover it. You don't even have to cover it. If it's green algae, it's going to sink to the bottom. If you have cyanobacteria, most cases where you have those gas vesicles, again, they're going to float up to the surface, and you're going to get basically your mini bloom within the jar there. So it's a quick and dirty way to do a verification test if you don't have high-tech tools. Next slide, please. So a little bit about toxins. I just show this, and this kind of leads to some of the problematic issues of working with cyanobacteria. On the left, I have some really common cyanobacteria that we see in this part of the region, and on the right, we see toxins that are produced by these. And Anabena, for instance, if you want to give it a click, Rachel, can produce all these different toxins. So if we do a toxin test, we see Anabena in our water body or dilutospermum, and we do a test for microcystin, and that comes back negative. That's a great thing, but that doesn't necessarily mean that there isn't other toxins that may be present within the system. So we definitely have to be careful. Click again. Aphanizomenon, that could produce a few different toxins as well. And click one more time. And oscillatoria as well. So you can have different toxins produced by the same genera of cyanobacteria, which makes it a challenge. So next slide. So how potent can these toxins be? And I must say, in the majority of cases, we don't see a lot of toxins being expressed in this region at high levels. But just for example's sake, these are, I'm showing the different cyanotoxins on the left here, and then I show on the right-hand side common toxins that we're well aware of: cobra venom, ricin, curare, cyanide, strychnine. And this shows the comparison of the concentration for the LD50, which is the lethal dose that will occur in 50% of the population in a microgram per kilogram ratio. So if we look at a microgram, just for comparison, a microgram is like a pinch of salt. Or I'm sorry, a pinch of salt is a gram of weight. A microgram is 1/1,000,000th of that. So you think of how tiny that amount is that can produce a toxic dose of 50% of the population. It gives you an idea how potent these can be. So for saxitoxin, for instance, you have 9 micrograms per kilogram compared to cobra venom, which takes 20 micrograms per kilogram. In anatoxin, which we call a very fast death factor, it's 20 micrograms. And this is often the toxin that is associated with dog deaths that you hear about in the news. From ingestion to death is usually under 20 minutes, so it's very acute and very fast, unfortunately. And there are just some examples of the other toxins as well, and comparatively. So they are something to be aware of and take note of for certain. Okay, next slide. Okay. Different toxins do target different organs in the body. What I show here is the different toxins on the left that can be found, and then what organs they target. Saxitoxin is a neurotoxin. It's found a lot in saltwater, marine environments, as well as sometimes in fresh water. So you have neurotoxins that you can get affecting the nervous system. You have hepatic toxins, which affect the liver. They can be tumor promoters. They've done studies in China around water bodies that were hyper-eutrophic or hyper polluted with cyanobacteria, and they've found a high prevalence of liver tumors in the folks that lived around those water bodies. They're known as weak carcinogens. Certain types can be dermatoxins. Depending on the sensitivity of the individual, they can be just cause rashes on the skin to actually some pretty nasty looking lesions and boils on the skin. It can be pretty severe for some. And some target multiple organs. So there's lots of different areas where these toxins can affect. Next slide. So routes of exposure that are important. You have the dermal exposure, the Lyngbia toxins, as I mentioned, that affect the skin rashes on this, effects from just exposure on the skin. Ingestion, this is the most common with pets because they're walking along the edge of the shoreline and they're wading in the water, and then they lick their fur. Everyone who has a dog knows they eat some pretty disgusting things along the waterfront. And when they do that with cyanobacteria, it can be pretty drastic. And then the inhalation, which is really common with the picocyanobacteria, which can be toxic as well. So you're getting that ingestion aerosolization of it into your system. Okay. Next slide. There's a lot of work going on at the molecular level with DNA and gene expression stuff with toxicity in cyanobacteria, and they still don't really have good answers yet. The science is still not there yet on what causes toxins to be expressed. We do know that usually in warmer waters and when there's hyperblooms that occur, we usually see more toxins expressed during that. But again, just because we don't have a visual nasty looking bloom doesn't mean that toxins may not be present in the water system. So it's been quite elusive for us, and we haven't found the real triggers that are causing a lot of these toxins to be expressed. So there's tons of research going on. That's certainly out of my realm of expertise, but hopefully eventually we'll start finding out why they are being expressed. And like I mentioned, they're not limited to the large blooming, large size cyanobacteria. The picocyanobacteria can be in low concentrations. There's work going on at looking at what might be the effects with chronic exposure to these very small aerosolized cyanobacteria. And there has been some associations with ALS that's occurred around the region where they're seeing if there's linkages between those picocyanobacteria, the aerosolization, and the chronic exposure. We also have blooms that can occur in small areas. It doesn't have to be lake-wide. And we also have some that can occur at depth where you may not even see them at all within your system. They're occurring down at that thermocline where the water stratifies. Next slide. So treatment options that we have, there's a lot out there. One size certainly does not fit all. There's a lot of research and a lot of marketing going on in this. You know, it's a big sales pitch for a lot of the stuff. There's the silver bullet, and I don't think there's a silver bullet out there. Some work for a while. Some work for a fairly long time. Some are just per event. You know, we do nutrient reductions along the watershed externally of the lake or the water body. That helps. But if you have nutrients within the system, no matter how much reduction you do in the watershed, it's not going to make a difference because the loading's coming from within the lake itself. It never hurts to help clean up the watershed and keep the nutrients out, but it's not going to stop the blooms from occurring. There's flocculants and binders. There's alum treatments that take place which bind up the phosphorus on the bottom. That can be a pretty good remedy in certain instances for a period of time. It's a management technique. It doesn't last forever. There's oxygenation and aeration to keep those bottom sediments from going anoxic and releasing the nutrients. There's ultrasound techniques out there where they put out these sonication equipment. A lot of drinking water suppliers use algicides, which can be problematic because you're actually killing the algae. It sinks to the bottom and just creates the nutrients that start another bloom to occur. Rooted plants can help. We've seen changes in rooted plants in a lot of lake systems where they've kind of disappeared due to manipulation of the hydrology with dams and outlets. People want their lakes clean. They want to get rid of these plants, nuisance plants around the edge. So they lower the lake level and it kills off some of these plants. Well, that's where a lot of your nutrient uptake goes in the summer. Trophic balances where you have something out of balance where maybe all your top level predator fish are gone and all you have left are the planktivorous fish. They eat all the zooplankton, so there's nothing to graze on the algae or cyanobacteria, and you have blooms because of that. Dredging is a treatment option which nobody goes for because it's completely exorbitantly expensive. Manipulations of flow and so on and so forth. So there's a lot of treatments out there, but you have to look at each one carefully and really weigh the costs, benefits, and side effects that might occur from it. Next slide. Okay. I just threw this in as a plug. If people are interested in reporting blooms around their lake and keeping connected, we have developed a phone app which you guys can use to connect with one another and notify folks of occurring blooms so it can be investigated. This was developed by us mainly because the states had that particular issue. People would call them and they weren't able to go out and investigate right away, or they'd hear about it a day later. And this was kind of for their benefit, but we thought it could benefit other folks as well. So it is out there if folks want to use it. Next slide. Okay. We do have a website called cyanos.org, which is our collaborative group, which anybody can join, anybody can go to and utilize the tools and information that's there, and contact us if you're interested in diving in more into monitoring your lake, kind of trying to keep track of it, kind of understanding what it's doing, what its characteristics are, how best to manage it, and that sort of thing.
That was really great. Thank you so much, Hilary. We have had questions coming in ever since you started speaking. So this is relevant to the treatments that you were just talking about. Are there any treatments in addition to what you already talked about that can be used to prevent a cyanobacteria bloom?
Other than what I just mentioned?
Yes.
Or,
I think that question has probably been answered, but I just wanted to make sure that it was addressed.
Yeah, that certainly wasn't an exhaustive list. I'm sure there's other techniques that are out there, and we're presently working with an interstate technical regulatory council that's building kind of a library of treatment options that are out there and their efficacy and cost and that sort of thing. That's in review now, but hopefully it will be out by next spring. That will be a useful tool.
Okay, thank you. This is a good question. Can these blooms affect well water?
I've heard that question before, and actually I only know of one study that was affected, that well water was impacted, and that was quite close to the water body. So it was probably under the influence of that well. It was drawing it in from the surface water body into the well. If you're not that close to the lake, I would say, as far as where your well is drawn from, you're probably okay. But I haven't seen a lot of research on that, and that's all I really know. There is a big concern in Maine where people are drawing it for the camps, and they're drawing it directly out of the lake itself, and that's definitely a concern.
Okay, thank you. Would ducks and geese ever be considered a factor for these blooms?
I don't know if I'd point the finger directly to them. Oftentimes these blooms that occur within systems, it's been building for years and years and years. There's a lake that I worked on that had a waste composting nearby, and it attracted thousands of seagulls that would come on the lake and wait kind of for lunch and come back. And I'm sure that had a nutrient input to it, but it's, you know, it's probably a lot more than just a few ducks and geese.
Okay. Monica wants to know, do these blooms affect just lakes and ponds, or do they affect rivers too?
Absolutely, they affect rivers as well, marine environments, both. And usually, again, it's the slower moving waters that warm up. So the river systems which are behind impoundments, or the really slow movers that have these issues.
Okay. Shay wants to know if the phosphorus coming in could be from lawn fertilizer.
Absolutely. I mean, fortunately, they're taking a lot of the phosphorus out of some of these, or the phosphorus out of a lot of these fertilizers. But anybody that does fertilize their lawn, you know, it's kind of frowned upon if you live along the lakefront, because you're not helping the system any. There are some that they can use that say are kind of lake safe, but I would be careful on the application.
Yeah, definitely. Kim wants to know if it's a large body of water, such as Great Herring Pond, is the entire lake or pond considered to be dangerous, or only the one cove or area where there's a sign of a bloom?
Well, if you looked at, if you remember that image I just showed with the kind of the purple and the fuchsia color and the blue on that lake, that kind of shows how sporadic it can be, and it can be really concentrated in one place and toxic and completely benign in another. You know, it's hard for someone to regulate that, because how do you shut down one portion of the lake and not the other? You don't know if the wind's going to shift and move it around, or if the toxins are released and in the water column. But yeah, that definitely happens.
And what about a question from Donna? What about fish and wildlife health when these blooms occur?
It does affect fish and wildlife. There's been plenty of studies done that have shown the microcystin toxin taken up in fish tissue and blood. So yes, it does have an impact. And there have been animal deaths. There's been plenty of deaths from these toxins of wildlife, as well as domestic animals and humans for that matter.
Emily wants to know if the toxins can be measured, and I'm not sure. This question might have been answered after she asked it.
Okay. Yes, you do measure toxins. I mean, there's plenty of tests, and you measure them in a concentration.
Shay wants to know, should we be concerned that this bacteria is aerosolized?
Again, it depends on the form. I'd be concerned if you had a lot of Cyanobacteria, they're really tiny, 2 micron or less size. But if your bloom is dominated by Annabina, or like I said, Annie, Fanny, or Mike up there, you're not going to have to worry about that as much being aerosolized. I mean, there is thought that maybe some of that might break down and you might get some toxins aerosolized, but I think that is relatively remote.
Okay. We have some questions here about use on the ponds, and just as a side note, I should have mentioned this earlier, Halfway Pond is also closed in Plymouth right now for a bloom, and that's a property that Wildlands Trust has about 400 acres of land around. So the question is, why are people still on the pond in speedboats? I will just say from Wildlands Trust, there's no speedboats on Halfway Pond, we know that, but we have signage posted all around our property, and we do as much outreach as we can. And I know that the Herring Ponds Watershed Association does too, but people still have free will. So Dawn, I don't know if you wanted to speak on that. I know you've been out actually talking to people who are on the pond and letting them know about the risks.
Yeah, we've tried to discourage people who do things like jet skis, people who are pulling children on tubes. Believe it or not, people were doing that. Jerry and I have been going out on the boat fairly often. If you're not exposed to the actual water, I'm not as worried about it. But if you go jump off the boat in the middle of the lake, obviously you're exposing yourself.
On a jet ski.
And on a jet ski, you're always getting splashed. So we've been advising not to use the jet skis, or if you do, to take it easy. But it's the uncertainty of the exposure, the allergic reaction that people could have to some of these toxins that's concerning, because you can't tell a priori who is going to land in the hospital for 5 days and who's going to be able to swim in the water for hours and hours without any ill effect. So we're not discouraging boats actively, but we're certainly telling fishermen. Most of the fishermen on Great Herring Pond that I went out and talked to, they catch and release, so that's fine. So no, I wouldn't say that you shouldn't go out on your boat, but I can't make that, legally I can't tell you to do anything.
Yeah, absolutely. Hilary, I don't know if you had anything to add in your experience.
Well, that's kind of the human condition. It's funny, I've been out and at boat launches and seen mothers with their kids playing at the water's front, and there's a big, huge sign that says, toxic cyanobacteria, keep out, public health warning. It's the human condition. Some people will ignore that and perceive it as minimal risk or no risk, and others will perceive it as a great risk. And my adage is always, it's kind of like swimming with sharks on the Cape. If you're 200 yards offshore and you're swimming laps or something, I'd be pretty risk averse to that. I'm staying inside the surf zone myself. So it's kind of a personal thing.
I'm just going to rephrase Tracy's question here a little bit. When would a pond be reopened? Like when would the town or the state or the entity that makes that decision have a test result that says, okay, we can open this back up?
Let me take that one because we're right in the middle of that.
Yeah.
The ultimate authority is with the state, but they will basically base their judgment on what the town says. So we're looking to the town. And what's the town looking to? The town is not looking to the amount of cyanobacteria, whether it's toxic or not. They're just looking to see whether there's a bloom.
Green color.
Now, whether, yeah, a green colored bloom. Now, whether it's a toxic cyanobacteria bloom anymore in our case, we don't know. The tests that we've had so far have been very low, and by low, I'm talking about 8,000 cells per milliliter. And bear in mind that 70,000 is necessary to close it down. So we're doing the runaround with the town about whether 7,000 or 8,000 cells per milliliter is enough to close the pond down. Now, I'm all in favor of erring in the direction of caution, because somebody might be sensitive at the 8,000 level, but it would seem that the presence of a bloom should not be the necessary condition for determining whether the pond should be shut down or not. Now, I'm up on my high horse and my pedestal here a little bit, preaching, but that's not the way it works. The way it works in the system is if they see a bloom, they shut it down. And that's what we have to deal with.
I'm back. Hello.
You're back.
I got kicked out this time. I guess it was my turn. How are we doing? Where are we with the questions?
I don't know. I saw one pop up that said, what's pigment concentration? I don't know if that's the most recent one or not.
That is the most recent question, and I actually cannot access the earlier questions because I was kicked out. So I have a new chat window. So if your question didn't get answered and you want to retype into the chat, go for it. But let's talk about pigment concentration.
Okay. Well, pigment concentration, like I mentioned earlier in the presentation, that pigment that is unique to Cyanobacteria phycocyanin has a direct relationship for the most part with the biomass and the cyanobacteria. So we can use that as a surrogate for cell count and concentration of cyanobacteria in the system. And it's a really useful tool for a number of reasons. You can go out there and directly measure. You don't have to send samples off and wait a week for them to come back and worry about some of the discrepancies in cell counting and that sort of thing. So you're actually just measuring that pigment concentration through the instrument, if that answers your question.
It would be really nice if the town and the state would accept those measurements, because Jerry has been doing them, and she's done measurements also on the samples over at the halfway pond, which is clearly 120,000 samples, cells per milliliter, and they clearly have a problem, and the ratio of the two pigments there is huge, almost 200, and our ratio is much less than that. So, but I don't know, frankly, both the people in the pond area and Hilary, I don't know if the state or the town Department of Health will accept that argument, because we are socked in with pea soup, but we're not getting those high ratios.
Yeah. Well, I think the state established this quite a while ago based on the World Health Organization's recommendations at the time, and it's is kind of stuck and they've established policy around it.
No.
So it's perfectly understandable that they wouldn't necessarily want to change. But that doesn't mean you still can't go out and measure phycocyan and pigment concentrations and kind of start to get a better handle on your system as it's happening and where hot spots might be in your lake or if it's uniform throughout your system, and maybe track the formation of blooms so you can get a jump on it if it does occur.
All right, so based on the testing to date, has it been confirmed that Great Herring Pond has cyanobacteria that is toxic, or are we assuming because some people have shown symptoms? Do we know the exact species? I feel like, Don, you talked about the exact species earlier.
Regardless of what we've heard in terms of the types of cyanobacteria that have been found, whether it's Hilary's identification, microscopy that Jerry has done, or whether it's been by the testing, they have been cyanobacteria that are toxic. We did not physically test for the toxins themselves. We tested for the type. We tried to identify the types of bacteria only. And it would have cost quite a bit more to have the chemical analysis done of the specific toxins. But the types of bacteria that we've seen, they are all potentially toxic. So we have to treat it as such. That part of the argument I don't have a quibble with.
Yeah.
Hilary, from what I understand, there's different toxins that can be produced as well, right? But what I understood was that the test for one toxin at a time, and so you could get a low toxin level, but be testing for the wrong toxin. Is that correct?
That's right. Right. You could just have one test for one particular toxin, and it might be producing a different toxin. So it's certainly not 100% potentially accurate on the toxicity testing. And they're expensive and time consuming. So, I think most people err on the side of caution without going through the expense of spending lots of money on toxicity testing. It might be a false positive or false negative depending on the toxin. Yeah.
Okay. Thank you. So Christine says that she has an old herring pond depth chart and it shows that the lake used to be much deeper than the current chart. Could the sediment or the lake getting shallower be a contributing factor to the bloom?
We've had a bathymetric chart recently done. I don't know which ones that she's comparing, but I know when we go out and do tests, we do them at different levels at the deepest hole. And the deepest hole, as long as we've been here, has 13 meters, 46 feet, pretty uniformly. So if there's silting in, it's not noticeable at the deepest part. That's the best I can do. But there is a more recent bathymetric chart that was put out by, the USGS survey. And you can find that on the web, and that's the most recent one.
Yeah, the one thing I will add to that I've seen in some systems is that through the years, and I suspect it's just kind of my gut, but I think some of this hydro manipulation with trying to keep recreational water bodies at a constant pool level has changed the bottom composition from a nice rocky substrate or sandy substrate to more of an organic, rich composition, because your plants grow, their growth phase is in the warmer summer months, and if you dewater in the fall, then all this plant matter is ending up in your pond. It's decaying and rotting on the bottom, and it moves into the closer to the center of the lake. So through the years, with this and dams have been in place in a lot of these systems since the 1930s and 40s. So this accumulation over time adds to the organic material, adds to the lakes. We've seen many lakes in the past 10, 20 years that have gone anoxic, that used to be clear to the bottom and rocky, and now they're covered with organic material and stuff. So not to say that's happening in Herring Pond, but we have seen that scenario.
Well, we do go out in the fall and take a rake and go in some of the less deep areas. There's plenty of rocks still there, but there's no question that we've been victimized over probably the last century and a half, maybe 2 centuries, by the cranberry industry that's been around just about everywhere in Plymouth. So, I mean, that's putting nutrients in and probably causing some runoff. We know we have a lot of runoff sites because we're basically a bowl-shaped water area around Great Herring Pond and around Little Herring Pond. So there's plenty of opportunity for runoff. If that, I may have misunderstood your question in my original answer, but yeah, I'm sure that there's some silt. We know that there's sand that's coming constantly from Little Herring Pond, propelled by the springs up there. So we know that we're getting that kind of silt. So yeah, I would bet that there's a silting, but it doesn't seem to be affecting, at least over the last 17 years that I've been here, the depth of that deep hole in the pond, the 13 meter.
The center.
So we have quite a few questions regarding safety. I think people are concerned because, you know, you might not always see the bloom. You might not know if the water is safe before you're entering it. So people want to know, how can they find out ahead of time about ponds that have been identified? And I will say mass.gov has a list of all of the ponds as far as I'm aware. And Hilary, you can feel free to correct me. The town of Plymouth also has on their website the 2 ponds now, Halfway Pond and Great Herring Pond, that are affected. They did have Ezekiel Pond on earlier this summer, and that has been lifted. And I guess, Hilary, if you could just kind of speak on, you know, what should people do if they get up to a pond and they don't know ahead of time, they haven't done the research, if it's safe or not, and there's no signage, should they assume it's okay, or should people just be kind of avoiding ponds right now?
You know, my personal perspective on it is real risk versus perceived risk. Again, if I saw a clear water body and went out in it and knew that there wasn't a big bloom that occurred a week before or 2 weeks before, I wouldn't be hesitant about swimming in it myself. I mean, you know, you can walk outside your house, you get hit by a brick, or a tree branch could fall on your head. You know, there's a risk walking outside the house, just like there's a risk swimming in the pond. And how you perceive that risk is really kind of an individual's choice. And I think common sense, being pragmatic, makes sense. If it's green and it looks yucky, you know, when in doubt, stay out. But I certainly wouldn't be averse or afraid of going into the ponds that look like, you know, they're pretty clean water bodies and stuff. Well,
and is the state website, to your knowledge, like the best place to go for up-to-date information about which ponds are closed?
You know, I'm not sure on that, to be perfectly honest.
No, actually, I think the Plymouth Health Department site is the best.
Okay.
Because the Clear Pond has now been downgraded. Clear Pond is closed, and that recently happened. So you can get that information from the Board of Health, and in fact, you can even sign up with them to receive information. Alerts as they happen.
So why don't I send the link for anybody who is Plymouth that's on this call? I will definitely send the link to the Plymouth website where you can find that information, but I'll also send the link to the state website for anybody who's looking for information outside of Plymouth as well. Yeah,
I don't think the state, actually DEP, monitors all the ponds for cyanobacteria blooms and that sort of thing. I think it does rest more with the departments of public health at the local level.
And then the landowners are responsible for any postings. So if you're a landowner with public access, like Wildlands Trust is, we're responsible for posting along our shoreline. And we also post at the trailheads to let people know.
Okay.
But if it's private property, a private landowner wouldn't necessarily be obligated to post.
Yeah.
Let me see. Oh, this is a good one. Will a cold winter help if we have a cold winter this year?
Well, not necessarily. I mean, we see blooms happen underneath the ice, but cold winters can't hurt if there's a lot of snowpack and stuff and ice. So yes, that definitely helps. If you look at ice out becoming earlier, it's just more time, more days that the lake is exposed to warming, warm sunlight and that sort of thing. Snow is a great reflector of heat, but without the lake's going to absorb heat much faster at a quicker rate. So yeah, good ice, that'll definitely help.
Okay. All right. And thank you, Suzanne, in the chat for including a link to the Six Ponds website, if anybody is in the Six Ponds area of South Plymouth. I think that pretty much covers everything. It's 8:35. I appreciate 41 people who are still on this call, and especially the Herring Ponds Watershed Association for putting this together. And Hilary Snook, thank you so much for this wonderful talk. It's so important to everybody right now to have this information. Is there any final words that you wanted to share with us tonight?
Just thank you very much for having me on, and I'd certainly appreciate any feedback if people got a lot of good information out of it. It helps me on the next webinar and stuff. If they found it informative, useful, they've heard it all before, that sort of thing. Be helpful.
I don't think anybody here has heard all of that before.
Pardon?
I said, I don't think anybody here has heard all of that before. That was really a lot of great science. Thank you for sharing that with us.
Oh, my pleasure.
I will be sending an opportunity for everybody to be giving feedback. It's just a short evaluation. It'll be part of the follow-up email. And Hilary, I'm happy to share that feedback with you.
Okay. That'd be awesome, Rachel. Thank you.
Yeah. All right. That wraps it up for us. Thank you, everybody. Have a wonderful night.
Thanks, Hilary.
And
Yes. Thank you, Don, Rachel. Thank you, everyone.